next presenter, who is no other than our own Andreas Gisel, an old member, young at heart, as you know, always, but one of the early members of EMBnet, who today is gonna give us a presentation on 3' tag RNA sequencing. Andreas, please.
Thank you very much. Good evening to everybody, and thank you to EMBnet to give me this opportunity to share some, oh, how do I do that? How do I share my slide here?
So there is a green share screen button.
Yeah, I see, but I'm blocked again here. I'm on a Mac. I'm sorry, just forgot that I have to set up my system here.
On the bottom, there is one very big green saying share.
I know. The security system is inhibiting me here. Okay, I'm sorry. Thanks to the EMBnet that I can present a small project I started some time ago with my students at IITA. This is to use the 3' tag RNA sequencing for gene expression study. The reason why we did this is actually very simple. Since we are working with a small budget and have a lot of RNA sequencing to do, we were looking for cheaper version of gene expression studies, and we popped onto this 3' tag RNA sequencing. Before I go into it, I just want to remind everybody how this works when you produce an RNA. We have DNA, we have the transcription here into the RNA, and we produce a pre-mRNA, which is composed of exon introns and has somewhere a so-called consensus sequence, which is important together with the U-rich region for a cleavage, which is more or less 11 to 30 nucleotides, although we observed different sizes downstream of this consensus sequence. After this cleavage and the splicing of the exons, we will have then the final product where we have the 5' cap of the mRNA and the poly A tail. So that's the first story. Just to remind you how this works with messenger RNA. Then we want to know what exactly is this 3' RNA sequencing. If we compare this with RNA-seq, as you see here, we have the RNAs with the poly A's. We fractionate this, and then we sequence all the fraction of our mature messenger RNA. The difference to 3' sequencing is that after the fractioning, with help of the poly A, we fish out only the fragments which are connected to the poly A, and we sequence only a short sequence of this fragment with the poly A. This would mean, in this case here, that when I have 4 transcripts, oh, I apologize, I'm jumping over in the wrong direction. When I have 4 transcripts, as I show here, I need 16 reads to describe with RNA-seq, whereas in this case here, the same 4 transcripts, I have only 4 reads. The advantage I have here is twofold. One, the advantage is that I have much less reads to sequence, and this drops my cost of sequencing like this. And the second, and this is kind of more the theory, is that each read represents 1 transcript. So I don't have to do all the normalization and the correction of all my biases I have from a whole RNA sequencing. So we did actually here, how it works is that we have our poly A RNA with a primer where I have a poly T. I fish them out. I remove, of course, the template, and I add with random primer my primer, the 5' primer, and I get my library to sequence. Oops, it goes always too fast here. After my purification, I can start my sequencing, and the important part of, why is this jumping here? The important part here is to see that I sequence from the opposite side than the poly A sequence. So I fish my fragment from the poly A, but I sequence from the opposite side into my fragment. So what is the effect of, has this effect on my data analysis? If you look here, that's visualization of full RNA sequencing. I see a major problem is my uneven distribution of the reads over the exon regions, which gives me problem to analyze this data properly. This is visualized by these graphics here, which it shows that if with a normal RNA sequencing, I have an uneven distribution of coverage over my transcript. And here in green is what I have, a single peak coming from the 3' sequencing. So with RNA-seq, I get of course, the full sequence, and I can find with certain applications, algorithms, splicing variants. This is what I'm gonna lose when I do this with the 3' RNA sequencing. The procedure we were looking at of analyzing this data, of course, is we are doing a local alignment using different mappers, mainly mappers which give me gaps or splicing sites, although I think we could also work with a simple bow tie. Then I will clean out the low mapping qualities. I will exclude, of course, the duplication. And then the major step, and this is what I want to concentrate on, is to see where the peak is, whether the peak is at my 3' UTR region or not. And this, as you will see in the further presentation, will be the tricky point. Once we figure this out, we can count the reads which map on this 3' UTR or close by, which then should give us the number of the transcripts. And if you have different samples, we can calculate the differential expression. So here is a sample of such a 3' sequencing. You see my gene here of interest. You see the intron exon distribution. And here at the end, at the 3', I have beautifully my peak, which makes me happy. Even if I look into at the end, where the peak is, in most of the cases, I find my consensus sequence, which should be the trigger of further down somewhere initiating the cleavage. But unfortunately, life is not that simple. Here we just compare the same sample we sequenced with RNA-seq and 3' sequencing, and you see again here the distribution of full RNA sequencing of my reads over the exons, and the big peak at the end here, which even goes into the UTR, whereas here we have a beautiful single peak. So the problem is now like this. Here is another example of a mapping of 3' of these 3' RNA sequences, and you see it's mapping here somewhere in the middle of my last exon. Question is my annotation wrong, or do I have some artifacts that produces me sequencing anyway? Looking into the downstream sequence, we find that we have areas which we have poly T which gets translated in the RNA to poly-A, which might cause a fishing with my poly-A approach, fishing fragments which are actually not really adapted, the added poly-A tail, but is a poly-A which is intrinsic in the RNA sequence. We have situation where we have two peaks. We have one peak which is nicely placed down at the UTR, but in this case we have a peak which is localized in an exon. Again, looking at this region here, we find again problems with this poly-T or poly-A's in the RNA. And we discussed with Benjamin from the University of Delaware some strategies where we search for A's, poly-A's with a sliding window and try to fish out these false positive hits to eliminate peaks which are not really peaks coming from the poly-A from the processing. Here we have a case where our peak is far away from any kind of a gene. So way outside of an annotation, in this case is about 15 kbs. We don't know whether this is an artifact. We don't know whether this is, belongs to another gene. Normally, the rules, what we apply is that if the gap is more than 5 kb, this could be an extended 3' UTR, which belongs to the gene upstream. So here again, we visualize this peak here. Actually, we have this peak also in the RNA-seq data set, and it could be that we have some non-coding or some small genes up there. This is another example where we have peaks all over. Most of them, I guess, are again false positive. The interesting one we were looking at is this one here, which is localized in an intron. And what is just to say in the beginning, this is a huge gene with a lot of intron exons, and especially this intron where we have this peak here is quite large. Looking at the RNA-seq data, we have quite a different distribution of these peaks. And what we conclude from here, looking more carefully into that peak in this intron, is that either we could have here an alternative splicing, or even in my eyes, more possible is that these are two genes and they are separated somewhere here, and this peak actually belongs to this part of the gene here, and this one here is a different gene, which one has to look into with RNA-seq data to get more information. So once we understood the problem, and we are still developing an algorithm to eliminate this false positive, I would go into the counting of these reads we were mapping on the genome. So we are using a high-sec count for this approach, and we were as first reading into the hits in exons. This is just an example here where you see always the highest exon, or almost the highest exon here, has the high counts, which is an indication that most of the peaks are at the right position at the end of the last exon. If we do the same counts on the UTR annotation, we see that this corresponds nicely with this observation we have here from the exons. If we do a count all over the genome, and I didn't mention this, we are working here in Cassava, which has a very relatively low-level annotation, and you will see this afterwards, why I say this like brutal like this. We have in total about 230,000 exons, and we count in about 20% of these exons, we have hits of these reads from these 3' RNA sequencing. Although if we filter out and take only hits, exons which have more than 20 counts, it reduces dramatically to 8%. Most of the hits, as we wish and hope, is of course in the 3' UTR. We have, with filtering, very low incidence in intron, and the important part is here. When we look at 5,000 nucleotides downstream, we have about 25% of these regions have hits localized down there. And this I can show with this small screenshot of an Excel sheet where you see the gene names here, and then you see on the first column the 3' UTR. If you have a 0 means I have no hit. If you have a dash, means I have no annotation. So you see, for example, in this case here, I have no annotation for a UTR, but I have a hit 200 base pairs downstream of the gene. I find hits. And the extreme case is, for example, this gene here. We have no annotation for UTR, but I have 1,500 nucleotides downstream of the gene. I have my hits of my 3' RNA sequencing, which tells me that most probably here I have UTR's annotation which are missing, and the gene is much longer than originally annotated. A last calculation statistics here I show you with this graph here. Again, I show you the different regions, UTR, 3' UTR, and then downstream regions we were counting. So here you see how many hits, how many of these regions have no hits. So we see, for example, of our 40,000 predicted genes in cassava, we have about 15,000 have no hits in UTR, but about 38,000, about 3,000 regions, 1,500 nucleotide downstream have hits in the in this region. So we see that we have quite a lot of 3' hits, sequencing hits, much further down that the UTR annotation would propose, which would claim that we gonna have with such a sequencing also a possibility to adjust the gene annotation in cassava. So the next step, of course, will be to do further analysis of these false positive hits of this 3' UTR, and then apply actually as a first experiment this expression analysis with this 3' tag sequencing in our experiments. So as a summary is that it seems a quite simple approach, the 3' tag RNA sequencing for detecting abundance. We see that we have a possibility to correct the gene annotation in cassava, and I guess also in other organisms. We can dramatically, and dramatically means 10 times and even more, reduce the cost for gene expression analysis. However, we lose information of sequences and also on alternative splicing. At the end, would like to thank the acknowledgement goes, and this is always a very important point for me. Doing good bioinformatics depends on the upstream procedure, and here we have people like Anatol, which is our trial manager, so he's doing all the planting, harvesting, and sampling. We have the people in the lab which are responsible for RNA or DNA isolation, and these are the bioinformatic people like Gisel and Tayo, which were responsible for the data analysis. Thank you very much.
Excellent. Thank you very much, Andreas. Wonderful presentation and a very nice, happy-looking team you got there. There is a question from Ina. Can we use 3' RNA sequencing in single cell level?
I didn't understand what level?
In single cell.
Single cell.
Yes.
I think there were some groups which were trying to do it, but I don't know the results. I don't know. Honestly, I have to say I don't know.
Okay.
Yep.
Okay, perfect. Thank you very much. If there is no other questions, and because time is pressing,
yes,
we should move on to our next speaker.